right, our next speaker is Dr. Renee Martin from the Medical University of South Carolina, discussing how to develop a plan for the assessment of safety.
Thank you. So hopefully what you've learned through all this, and anybody who's met with me probably has really learned this, is that as you endeavor any of these things, you end up with more questions at the end of the day than you came into it with, right? So I think in thinking about safety, and I've talked about safety with a number of you, if you know me now, I'm going to ask you a lot of different questions, and in that forum, it's really hard. Then there's not always a right or wrong answer, and so it's very much a balancing thing, and that's hopefully what we're going to kind of see today. And I'm going to discuss different things with this. So let's just say we start off with these specific aims. You want to demonstrate that your drug is better than the control at improving outcome, or you want to demonstrate that your drug is safe. Do you see anything that might be not so great with these specific aims? Anybody speak up? Come on, let's make this interactive. This is how I teach, right? Yeah, how can we prove it's not safe? How about even the first one?
But it's better.
Perhaps clinical equipoise might not be there if you already know that your drug is great, right? You're trying to demonstrate it that it's really good, right? So this is just more semantics, and we have to think about how we actually state these things. I think this has consistently been talked about throughout this course, but the one I'm going to focus on is the second, obviously, because how do we prove something safe? That's actually really hard to do, especially in this realm where we're starting off with these earlier phase studies, and we have small sample sizes, and the likelihood of even observing some rare events may not happen, and so it's really hard to prove safety. The fact of the matter is, and if any statistics class you've ever come with, at the end of the day, let's just say you don't have enough evidence to reject the null, right? I don't have enough evidence. So is it truly that your drug is not effective or whatever, it's not real, or you just didn't have enough power or whatever? So I kind of try to summarize it here. If we have insufficient evidence to reject the null hypothesis that your drug is safe, this does not prove that it is safe, kind of thing. So it's very difficult. More likely, a lot of times what we're saying at the end of the day, we didn't see things, well, there were no safety concerns identified. It doesn't mean that there were none there. And this definitely has a lot to do with the number of patients that you actually have seen in your study. So we need to be clean and clear about the specific aims, especially when it comes to safety and how we're stating these things. How you're measuring safety, that's one of the things we'll talk about, and I've talked about it with the people that I've consulted with, exactly how you're going to quantify this. That's one, you have to really spend a lot of time with it. Safety itself is not an outcome, right? So we want to think that. So maybe what we really need to do and really change the frame of mind is we're trying to identify harms, but we're not going to prove safety in that process. Another key thing is that safety is a part of the whole life cycle of drug development or whatever you're doing. A lot of times we might say safety is focused in those earlier phases, but we still see things, especially in the later phases when your drug or device is now being administered to a broader population than it might have been in these earlier studies. So, all of a sudden we're seeing new things. And again, or we're just more people are being treated so we can see some of these rare events. So the one I like to share, it's not a good experience, but if you're familiar with the ALIAS study, it went from early phase to phase 3, and I was the unblinded statistician in that study. And the first DSMB report I had to produce, I got a little scared because there was a differential in these death rates. We didn't see anything like that, obviously, in the earlier days. And as time progressed, it got worse and worse. We ended up having to halt that study, redesign it, to then move into ALIAS Part 2. So it's a matter of, okay, well, there were only 2 sites in the earlier study, right? And now we're, multiple sites, few patients in earlier studies, few patients at the different doses. Now we're at our dose, we're treating more patients and we see things. And this is not uncommon that as we move along the process, you start seeing other things that potentially may show up. So you really need to think about what things are out there, and we'll talk about that. Unlike efficacy hypothesis, safety hypothesis sometimes can't be pre-specified. You can think about certain things, and as you move through the development of your drug or the process and stuff, you may have particular things in mind that you can specifically say, but they can be more difficult. Again, I talk about sample size here, but most studies are designed for efficacy. They're trying to find this efficacy outcome, and so that's where they're powered. A lot of times, our safety outcomes might be rare events, so we're not powering to detect rare events in this case. So it's just one of those things to consider. But where does all of this come from? Chris talked yesterday about the development of the earlier designs, this CRM, the 3+3. And a characteristic thing in this is that you're going to treat a very small number of patients, 3, very small, and you're looking, okay, just 1 or 2 actually have some safety event or toxicity, and that kind of determines how you move forward or back and stuff. And the key thing here in cancer, typically there's a high frequency of toxicity. I mean, you're looking for a higher level. But that might not work in our field. I mean, let's just say our safety event is symptomatic ICH. 30% rate of symptomatic ICH, that's just not going to fly, right? We're not going to ever accept that. So we're in a different realm, and so it's just more difficult to do this. So you really need to think deeply about, is this a new medical device, drug, whatever? It's just right on the market. What stage are you at in this? So, what is that target or mechanism of action? These are the kind of things you kind of have to think about, explore, and then from that, really try to nail down what can I expect as far as safety events? What do I think? What might be that expected rate that we want to see? So maybe there's an investigator's brochure that kind of will give you expected adverse events and what those rates might be that you would think. There may be other studies of this drug in a different disease area. We've been exploring some of that in our group. So this drug's been used in a different disease area, and I want to use it now in mine. So maybe I can think about what adverse events I might have with that. But you have to be very careful, because a key thing here is to be mindful of what we would expect in relation to your drug, and what you expect in relation to your disease, and how you want to tease those out. I think I met with someone a couple of days ago, and it's like, okay, you're going to introduce something new into this process, right? But we have all of these other things going on, and how are you going to tease out what's due to your new intervention versus all these other things that are going on at the same time? And it's really important, because that relationship, you want to be able to kind of say what's expected in this disease population for this, because we have to come up with what we expect in our control group, right? For those untreated patients with my intervention, what do I expect? That way I can compare what the rate is in my treated patients and kind of have something to go by. So, what do? You go to the epidemiologic data or natural history data to find that, or see what you would expect in your control group, or maybe a control group of similar patients. So these are all things we kind of need to have in place before we're even developing our safety plan, right? Because we need to understand that. So let's just say you have a really good idea what you think is going to happen in your control group now. So now, how much of an increase in this is going to be cause for concern, right? Is this very dependent on the disease area? There's no right or wrong answer in this. It's going to be what's clinically meaningful and acceptable given that situation. One example is a relative risk of 3 or more. That's one criteria. I've talked to someone, if we have a certain rate, sometimes we just look at the upper limit of the 95% confidence interval and say, if you're beyond that, that would be extreme. That would be, that's one criteria we've used in studies. The whole idea is you got to come up with something that you're comfortable with and something that is a trigger to say, okay, this is cause for concern. And that helps you to define your safety analysis. Another thing we need to look at is the risk-benefit ratio. We may be in the realm of cancer, you may be more willing to accept higher levels of toxicity in that short-term period because of this risk-benefit ratio. However, let's just say we're doing a prevention study for recurrent stroke. You're going to be treating this patient for a long period of time. The actual risks at baseline are either low or moderate, they're not very high. And we surely don't want to introduce more problems for these patients at that point. So perhaps that risk-benefit ratio, it's going to be quite different than what we would be doing if we were, accepting if we were in a cancer study. So no right or wrong answer. It's going to be what fits for your disease area. So switch a little bit, because we've had this safety and tolerability. A lot of times they're kind of thrown in together, right? But tolerability is actually kind of a different concept, right? So here we're really talking about, can patients stay on the drug, or receive the whole treatment as it's intended, right? And so sometimes we're saying we're at a phase where we really want to know, is this tolerable? So we have to come up with definitions on what is tolerable. So an example, so if 30% of patients stop taking this drug due to minor side effects, you may have a tolerability issue there. We want our patients to take the drug as is intended. So you need to, again, have some type of way to quantify this. And I think this is probably one, if safety is not hard, tolerability is even harder, right? Okay, what's acceptable? Is it 60%? Is it 10%? Wherever you're drawing this line on this, you have to come up with criteria. Where this is hard is sometimes there are ascertainment issues as far as tolerability. If this is a drug, it's a prevention trial, it's something that they're taking over a span of time, right? So are we going to use pill counts? We can be innovative and say every day go put in your iPhone that you took your medication. That's, this, you're still relying on this self-report a lot of times on where they taking the drug, if it's that kind of thing. How is this all affected if there's a dose reduction or starts and stops in this? And how are we going to do this? How are we going to quantify it? Is it the number of days on drugs divided by the days expected to be on the drug? This kind of thing. So there has to be a quantifiable definition, and we have to be able to collect that consistently with good quality. Okay, so we kind of talked about safety and tolerability. I'm going to go back and focus now on safety. So we want a safety objective. We need to identify harm, but we're not proving safety. So how do we do that? Here are the steps, kind of like we kind of just went through. So we want to anticipate those potential harms, define that primary safety outcome. It might be multiple safety outcomes, but what we want there. I say here a composite of several potential events, and we'll look at that in a few minutes. And then we need to know what these expected rates are in our control group, and then how much of an increase are we going to be concerned about to say, oops, we need to do something, or we just look at that data. So how do we measure safety? We measure safety via adverse events. And I have the standard definition of an adverse event here, and it always surprises me how this definition is not universally known. So an adverse event, any untoward medical occurrence associated with the use of a drug in humans, whether or not it's considered drug-related. That whole part, the whether or not drug-related, because I can tell you a number of times I'm consulting with a clinician, it's like, oh, we're just looking at things related to my drug. Well, no, adverse events encompass everything that's there. And we maybe focus on specific adverse events related to, but sometimes it's hard to tease out that relationship. And so we need to make sure we can understand that and identify that. The other part about adverse event collection is that it's passive. So your patient comes in, so what have they observed in the last 3 weeks or last month, this kind of thing. So it's always kind of like after the fact. It's anything that's kind of like in the medical record or that patient's reporting to you that they've experienced, right? Well, all adverse events are recorded after randomization. That's another thing, because we have pre-existing things, and we don't want those considered. When a patient comes into your study, it's at the point of randomization and forward. Typically, we are coding these using some type of coding system. We use MEDRA mostly for our coding of adverse events. And with MEDRA code, adverse events can be coded into body systems, preferred terms, higher level terms. So there's a hierarchy of how these, one event is going to be categorized throughout the process. And then by using this, we can come up with a cumulative occurrence rate by treatment group that we can then present to the DSMB. So here, it's kind of hard to see probably, but here's a illustration of how we might present this cumulative, this aggregate data on our adverse events. I really want to go up with it. So this is a study where you have 3 treatment groups, A, B, and C. And on this table, not only do you have the number of these events by body system in preferred terms, you also have by severity, and you also have the percentage of subjects with that adverse event. So different ways we can summarize this, because just looking at a number doesn't really give us a good feel if there's something going on. I want to point out in this that, because it's not in my slides, but there is severity and serious are 2 different things, right? All adverse events are rated by severity, and then some adverse events are serious or not, and there's a definition for that, and we'll get to that in a second. But this is just one illustration of how we can look at this, and if you think from a DSMB's perspective and you're presenting them this kind of table, so what's going to be alarming, that it might be really hard to tease out what are potential safety things going on in this case. So, but it is one way to quantify it. But we also have issues with MEDRA. So we have issues with how adverse events are recorded. So sometimes it's not the actual events. We have all the signs and symptoms related with that event that they actually get reported. And so instead of having a single event, we have a multiple list of different signs and symptoms. So one of the things we do in the data management center is try to see what adverse events are reported and try to help improve the quality of that reporting. Say perhaps this is just one thing versus a multiple cascade of signs and symptoms for a certain event. So that's one thing. Body system, sometimes it's too broad to look at it just by body system, right? Because a lot of different things can be grouped into one body system, and so this might not be the best way of identifying a potential harmful things that we are at some type of signal there. The other part with MEDRA is that different, very related terms can actually be mapped to totally different body systems. So by body system, it might not be the best way to capture, like, okay, this is really a problem area. So it can be hard to detect safety issues in that case. So what do we do? So first of all, we have to start with the reporting of things and make sure we're doing this consistently with data collection. So that's the first step. The second step, and I've used this probably in almost all the trials that I'm involved in, is we're not looking at a particular event always. Sometimes it's a group of events. So we may group events to say anything that falls in this, create this composite. That's what I want to identify. So we create, those are special safety events kind of thing that we want to look at, and that's something we specifically monitor for all subjects, and I'll talk about that. So in essence, rather than just relying on AEs being reported like this, if I'm concerned with pulmonary edema and cardiac arrest, I'm going to say, did this patient have pulmonary edema and cardiac arrest? I ask that question specifically, because remember, adverse event reporting is passive. So if I don't specifically ask for that, I'm relying on someone to report it to me, or it's in that medical record, right? So, so I'm going to show you a table in a second, but for the Alias trial, we had particular safety events that we were interested in. So we asked that specific question every time that patient came in. Did they have cardiac arrest kind of thing? And so Right, so it's just a different way of asking the question so that I can get the information I want, and I know that for every patient that question is asked. So it's the difference between a passive and active way of collecting the data. So, like I said, one thing is to use the grouping of terms, and the other is the way in which we actually ask those questions. Here's just a simple illustration about this grouping versus separated things. So, I mean, it's a very simple thing. So if I'm only looking at the individual events here, and I'm comparing treatment to control, I may not be concerned maybe for constipation, and that's the only one where it's a differential of 3. But if we actually say, I'm going to group all of these together, and this is a composite event, then I have a rate of 8 to 2, and that might be much more alarming. Okay, so we're going to consider these are all separate events and separate subjects of things. But it's just that point to illustrate that sometimes it's the grouping, and we know that a single adverse event might not be as interesting as a collection of things that actually are very related that are associated with this disease or this drug that we might be interested in. Okay, so I actually got ahead of myself. So remember, it's prospectively collected. So if you're interested in a particular safety event, one way to do it is to ask about that safety event every time you're seeing that subject. So one way we kind of put it on for our studies is we know that our adverse events are collected at certain time points, and it's on your data collection schedule. So we may have questions at the top of the adverse event form that are our specific safety events that we're asking for. So we know it's asked every time you're actually collecting adverse events as well. Okay, so the difference between an adverse event and a serious adverse event, and this is based on an FDA definition. So a serious adverse event is any adverse event that's fatal, life-threatening, results in hospitalization or prolonged hospitalization, results in disability or congenital anomaly, requires intervention to prevent permanent impairment or damage or other important medical events. So it's different from severity. I just really want to continue to say that. I teach a methods course every year, and I ask them to summarize serious adverse events, and I can promise you every year I get people giving me severe adverse events, and that's not what, so it's different. But sometimes, we don't want to just look at serious adverse events. Sometimes when we're designing our studies, we might be looking at all adverse events during hospitalization, but just the serious adverse events after hospitalization to 3 months, if that's how long we're following them. So we do use different criteria for collecting these things, and just depending on, again, the severity of our patients and what we're looking for. Okay, so we want to develop a safety analysis plan. Well, safety definitions and safety analyses can be found in a number of documents that we use throughout a study. And you may have all of these documents associated with your study, maybe a few of them, but these are the typical ones that we would have in there. We'd have a safety monitoring plan, which is different than a statistical analysis plan, which is different than a DSMB monitoring plan, which is different than the formal plan for pre-specifying our interim analyses and stuff like that, and which is different than how we're reporting things to the FDA or the DSMB, which is different from these expected adverse event rates, right? These are all different things, but all of these pieces and parts are associated with safety. So you will have safety and safety analyses kind of intermingled throughout all of these things. All right, so what's the difference between reporting and summarizing our data, right? So we report things to the IRB and the FDA, and there are guidelines for what needs to be reported when, depending on the type of study that you're running. But the group, the body that actually looks at your safety data in aggregate by treatment group is the DSMB. So that is the one body who can give you, can see the full view of the safety by treatment group, and that by treatment group is important because if you're a blinded study, they are the group that are seeing things by treatment group. No one else is. So who's watching safety? So as an investigator, you can see things on a patient level, right? Because a blinded study, you don't know what treatment, but you're seeing what adverse events that patient's having. The clinical monitor might see the safety at an individual level for over several sites. Your medical monitor, still blinded, will see what safety events as they come in, one at a time. Again, the FDA, they'll get annual reports, they'll get SAEs in real time. IRB will look at those SAEs at their local sites, if it's in that frame. But it's only the DSMB that sees the aggregate data, all of it by treatment group. So planning safety and safety analyses for the DSMB is a really important thing and something that you have to think about ahead of time. It should clearly describe the details of the proposed plan for interim data monitoring of the safety events. What's going to be monitored, what endpoints, what AEs, the timing of this, the frequency of data reviews, and then criteria that guide early termination. This is a hard one because we have to establish criteria, like I said, they're looking at the data, there needs to be some trigger to say, what's going on, right? Especially for things that are unexpected, or if, you start seeing differentials and things, but it's, how do you define that? It's not always clear, and so you have to come up with something that's reasonable, and this is something that's done a priori, so you want to really think out what those guidelines are going to be for them. So this is a good question. Should the DSMB know which treatment group is which? Should it be totally unblinded? If it's a blinded study, should they know this is your drug, this is placebo? Depending on the institute, some institutes, the DSMB is always unblinded from the get-go. I think most of the studies we run through NINDS, the DSMB will start off partially unblinded, AD, they know AD, but they would always be given the ability to be unblinded, especially if there's cause, or at their discretion. So the other thing that has to be stated in your safety plan and stuff and how these things are done is about this unblinding, when it will happen, how it will happen. You think about those things. And know, as the investigator, you may or may not know if the DSMB becomes unblinded. There's a lot of information that is going to happen. You plan for it, but you don't necessarily know if it indeed happens. And typically, less knowledge is better. It's kind of the philosophy I think we've kind of run down. But you want to think about that. All right. So we kind of talked a lot of bits and pieces and questions and stuff that have to be answered. What about the safety analysis? What do we need to think about? Well, a big thing is who's included in the safety analysis, right? So we talk about an ITT analysis for efficacy, but is that necessarily the best sample that we should use for safety analysis? Probably not. It's, we want to think about who's receiving the drug, and those are the people that are at risk, because if you didn't receive the drug, you're not at risk for that, that safety event. So we typically define a safety sample, and we put it in the protocol. Safety sample, let's just say it is a prolonged treatment. They've received such and such % of the study drug, or as long as you were on study drug, you're part of the safety sample, that kind of thing. So you need to define that. And the other thing that's in here, crossover should be analyzed according to what they actually received, right? So let's just say we have a medical management and the surgery arm in our trial, and this someone randomized to medical management actually has surgery. In my safety analysis, do I really want them in my medical management arm? I would think I would want them in my surgery arm, because they received surgery, and I can truly evaluate what that's happening. So the fact of the matter is, what you need to do is think about these things and spell them out a priori. That's all I'm going to be saying most of this talk, is that you gotta think about it. Okay, so now here's the steps, and now I've added one more to that step that you need to think about. So think about specifying your sample and your sample size, because again, we're not powering a study to detect safety concerns, right? We've powered it pretty much for efficacy, so what do we do? You want to think about that. Think about what power you would have given that sample size for especially potentially rare events that you've considered are rare safety events for your study. Other things that you want to think about when you're defining your safety analysis is, do you want this to be a two-group comparison? Do you want to compare your treated group and your control group in your study? Or perhaps maybe you have these expected rates which you think and you just want to compare to this historical rate. You know, which one of those? You just have to specify what you're using there. Do you want to do one-sided or two-sided tests? You know, if you have two active treatments as in a trial, perhaps you want a two-sided. But what if it's control placebo and treatment? Maybe you just want to see what's worse on the treatment arm. So it's one of those decisions, one-sided or two-sided. Rare events, what are you going to do with that? How are you going to handle that? You got to think about that ahead of time. What do you want to do with that? Other things to consider, so one or two-sided test. Are you going to report confidence intervals around that effect? A lot of times we do that, especially as we're looking at safety along the way, because as we have accumulating evidence, our estimation is going to become more precise, hopefully, should. And so typically we like to provide confidence intervals so we can give a feel to the DSMB for that precision of estimation. How are we going to quantify them? Is it the frequency of events? Is it the relative risk? Is it absolute risk? Is it observation? I mean, different ways we can quantify that thing. If we're doing time to event, we may be reporting hazard ratios. And then when we're doing safety analysis, do we want to adjust for these baseline covariates that were important enough that we put them in our randomization and important enough that we put them in our primary analysis model? We pre-specified that. Should they also be in the analyses, or should we also adjust for them in our safety analysis? These things just need to be stated a priori. So obviously you're consulting with your statistician to decide these things, but these are things that you should think about. The other thing we commonly have to think about is, our DSMB is going to be looking up this data repeatedly throughout the study, right? I've had studies where I was producing DSMB reports every month. So that means 12 times a year my DSMB was looking at my safety data and these things. So do I take, do I have issues with multiplicity? Will I increase the likelihood of finding, a statistically significant difference, even if none exists because of this repeated looking at the data? When we're thinking about efficacy and from a frequentist point of view, we actually have to adjust for those things, right? The number of times we look at the data. So it's something that you want to talk about and think about. This is an example from a trial, and what we can see here, these are estimates over time, and what we see here is at that first look at the data, you have, pretty, looks like a pretty large effect, but the confidence interval on that is pretty wide. But as we accumulate evidence throughout the trial, you can see this kind of goes, it's regression to the mean kind of thing. It's getting closer and closer, and our estimation is getting tighter and tighter. And so it just is what it is. Sometimes you got to be careful of looking at things on a random high, especially at the beginning part of a study, because we have fewer patients and we have a lot more variability in the data and stuff. So again, something to think about. So should we got to remember we're not trying to prove safety. So we're just trying to quantify risk. So multiplicity is a little less of a concern if you're worrying about it. We just need to think about if you want to make adjustments for that kind of thing. But we want to identify harms. So we're going to be looking frequently at the safety data. It's often difficult to define those boundaries for what's concerned, right, with for safety. But you can depend on experience sometimes to help guide you as far as what those boundaries can be. You want to develop stopping rules, or you have to decide a priori, do I need to have formal stopping rules for my study? This is an example, let's just say the expected symptomatic ICH rate is 3%. And if you say, now we're observing 6%, is this cause for concern? Do I need to have a rule in there to make, to make sure that we're not putting patients at risk with this treatment? And that's the big thing here. But the key is that these stopping rules should be specified in advance, should be written in your DSMB monitoring plan, in your statistical analysis plan. They should be found somewhere. They're rules, they're guidelines. Stopping is not necessarily mandatory, but it does help. I can tell you as a DSMB member, it really is helpful to have these there so that we are not reacting to something, because when you see something you don't expect in a report, especially when it comes to safety, this could be the generation of hours of discussion within a DSMB to say, what do we do? I've been a part of this. It's not fun. And so you want to have some guidelines there. Monitoring, it does involve a combination of statistical and clinical insights. You're not in a deliberation of a DSMB, right, when there's a safety concern there. Typically, there are clinicians who are experts in the area that are there, and those experts are being, they have their perspective. The statisticians are kind of trying to help in interpreting the statistics that we're seeing in realm of things. So it takes a combination of this to really discern, is this a signal that we really need to act upon? But the key is that you want to stop if the trial poses unreasonable risk on your patients. And so we have guidelines for that. Someone's going to make a decision on that. And so it's one of those processes that you have to go through. Here's another example of how we have presented data, AE data. And this is for specific safety events. This actually comes from the POINT trial, I think, where you have specific safety events that we were interested in. What you see in this table is the expected event rate. So that you imagine this is a table that's in a report that the DSMB is reviewing. Okay, so what's in there is this expected event rate that we had stated a priori in the protocol or whatever. It was set out from the get-go. You have this accumulating data, so all these fields are being populated now, where you can look at the number of subjects at risk for that event. You can look at the number of events. You can look at the event proportion. We present a relative risk and 95% confidence interval on that. We can look at the event rate and the 95% confidence interval on that. So we're given a lot of information to then judge, okay, what we're observing in comparison to what we expected, to kind of weigh that risk-benefit ratio. Like, is this something for a woman? Here's another way we can present the data. And I'm presenting ways you can present the data because we want to think about this, because you're working with your statistician to develop the templates for these reports and how we're going to actually report data to the DSMB so they can make the best decision that they can. This is another way of doing this where we have, I think this was kind of a model that we were going to use or may use for the most trial, where we have 3 treatment groups and maybe we have age strata here. And we know what the expected rate is, or it could be 3% or 5%. And what we're doing is giving the probability, this here, of observing what we see, or the probability of seeing what we see, given that this is the true event rate. So extreme probabilities, things less than 0.5 might be cause for concern. And it would be a trigger to say, perhaps let's see what's going on in that strata or that treatment group to kind of look at things. So it's signals. This next is another. It's now we still have event proportions here by treatment group, but now on the right side you have you can put the risk difference. We could put a hazard ratio. So in essence, you can judge where patients are benefiting or being harmed by the treatment versus the placebo, and get an idea on where we look at things. We really try to provide graphical illustrations of these statistics, because a lot of times it's a lot easier to see things than just judge by numbers. So we try to be creative with this. This is a volcano plot. It's another way of looking at this, where we're looking at the probability of observing these events given specified event rates. And we're looking for extreme probability. So here in this area, we can see things, our volcanoes blowing up. We can look and see what things are coming out above, beyond what is expected, and then investigate what's going on there. So just examples of different illustrations on how we can look at safety. All right, so this is for adverse events and stuff like this, but what about labs? I know for one, I wasn't trying to be too critical, but when you start saying I'm looking at safety via my lab data and vital data, I'm like, that's a lot of data. How are you going to look at it? How are you going to quantify it in a way to say, this is a safety signal? So we really need to think about that part of it, especially given this kind of data. So I have some illustrations of things we can do given this type of data. So we might look at box and whisker plots. That's one way we kind of look at things. And especially if it's collected over time repeatedly, we can look at this, and I'll show you an example. Other illustrations may be a shift table or a heat map. So let's look what those look like. So here's your box and whisker plot. You can see this is data collected over time by treatment group. We have A and B represented here. I mean, if I'm just looking at this graph right here, it looks like all the means are about the same, right? Over time, I'm not seeing any differential between the treatment groups, but I don't think it's mean lab value that we're ever really interested in if we're trying to identify safety signals, right? So what else you have on this graph, and you might not be able to see this in the back, is the normal levels. So there's a range of that. So what might be more important is to see where we have extreme values and see if those extreme values are differentially, separating for the treatment groups. So the ones that are beyond our normal limits kind of thing. One way of looking at this. Another is a shift table. So we have data collected over time, and let's just say what we're interested in is patients that are going from normal to abnormal, right? Because that's a concern. So how are patients shifting differently for the treatment groups? So that's what a shift table can give you. Here's an example. It's kind of odd. So if we look here, we can compare treatment A to treatment B, and the percentages of those that are shifting from normal to abnormal, and make a comparison there via a shift table. So another way to illustrate these things. The other way we can do is a heat map. Heat maps are easy to look at, really neat. And so here we're kind of looking at over time what things are actually going beyond a certain limit. So in essence, we're taking our continuous measure and we are categorizing that and then quantify it via some type of illustration that you can look at. So this is an example of a heat map, and what we can see here is the reds are the high alerts. These are the ones we really want to be concerned with. But what we see in general is our creatinine group actually had more things of concern. And it's an easy way to just quickly look and see if something's maybe a signal. And so, again, with this type of data, labs and vitals and stuff like this, there has to be some way to quantify to say, this is okay, this is a safety concern. And we use illustrations to kind of try to point out those things, and these are some examples. What about unexpected events? And this always happens. You can't anticipate, they're unexpected, so it just shows up. So what do you do in that case? It depends on the balance of risk to benefit. It depends on the severity of the AE if this is unexpected. Typically, these are the kind of things you want to make sure that are pointed out. Like, we don't expect these things. What do we do? A lot of times when these are identified in a DSMB form, we really start looking into the details of those specific events and try to, without alarming anyone, just try to really nail down what's going on with that. And why does this happen? Well, we already said this, sometimes this is typically with rare events. Lots of times we have small sample size, so it's not going to be till we have more and more patients treated that we actually might see something like that. A lot of times in earlier studies, we exclude, we're very exclusionary in our studies, and so it's not till you get to maybe the phase 4 where more patients are being treated with this, and all of a sudden you're seeing these unexpected things. These types of patients weren't necessarily included in your earlier study. And then you have to be careful, again, with how you're defining that safety standard. What are you using your denominator? Like, are you using the number of persons at risk or person time? You want to make sure that's clear. So in summary, know what's expected about your drug. Spend a lot of time doing the research in that. What's out in the literature, what other disease areas may have used a certain agent or this product, whatever there is. I would say ask as many people who are related to the area as you can, because you never know what perspective they're going to bring. I think I asked a lot of questions yesterday, and I created a lot more problems yesterday than I answered, but I think especially talking to a statistician who is naive about your area, sometimes we ask a lot of questions, right, that kind of bring things, why didn't you think about it like that, right? So I think that's a valuable thing. You want to, and from that, pre-specify those adverse events that are important. Create composites and your safety events that you're really interested in. Consider that risk-benefit ratio. Come up with a criteria by which you're going to act on something, or at least advise your DSMB to act on if it's beyond a certain value. And this is my final slide. This is my little dog. This is Ruami, and she actually goes to college with my daughter, Jean-Marie, so they're enjoying life for a better time. So that's it. Thank you.